My second student is Austen Stoelting. I hope I pronounced it correctly. She told me the E is silent, so we learn. Also, she comes from undergraduate here from JSU, and she's a medical school type of student, and hopefully we will send her to medical school very soon. She came to the lab last year, and I told her there is no compromise with the thesis. She has to do a complete thesis to graduate within a year. She said she's willing to work very hard, and so far she proved me right when I told her you are a very good student and don't sit at home waiting for the graduate school to be accepting you in medical school. We could provide you with the knowledge in here. We could make you a better person too. And she agreed with that. She's been working extremely hard. Within the last year, she collected the data, and she will still working on her data. But her proposal being complete, that means part of her thesis be written. Today, she's going to present here most of her data, but not complete. And in her work, she examined the interaction between the symbiotic interaction between selected fungus and virus with the tomato plant. Consider this a new research and a lot of emphasis right now on drought and trying to find a solution for the drought. And she's going to cover that in her talk, and I hope you will enjoy it. Okay.
As he said, my name is Austen Stoelting, and I've been working with Dr. Hamdani for quite some time now. I'll be telling you about a symbiotic interaction that we've been working with involving a tomato plant, a fungus, and a virus. Okay, just to walk you through my presentation, I'm going to cover some background information, tell you about our objective, hypothesis, materials and methods we use, and then our results and conclusions. Okay, as we know, temperatures have been rising in the United States, and the number of heat waves and regional droughts worldwide have increased. This figure shows a map of the United States into 2080, and it shows that the number of years between extreme heat events is decreasing, meaning that they're happening more frequently. Okay, obviously droughts are devastating to farmers and those who rely on their food. This figure shows a map of an intense drought in 2011. It shows the severity of the drought, especially in Texas. You see the darker reds in Texas, which caused over $5.2 billion in agricultural losses. So far, I've only mentioned the United States, but regional droughts are increasing worldwide. And these are especially affecting poorer countries because they have limited resources. The problem can be solved, though, I believe, through the application of science, although I don't think Farmville falls under this category. But this is not a new problem of drought. Over thousands of years, mankind has attempted to find different solutions with questionable results. Okay, but there are solutions to drought. One such solution is irrigation, although this can be difficult because water resources are very limited. Okay, so far I've talked about humans' responses to drought, and now I'd like to move towards plants. How do they respond and deal with drought and high temperature? One thing they do is establish symbiotic relationships with fungi, for example. Previous studies have shown that this particular fungus, Curvularia protuberata, can confer heat tolerance and drought tolerance to tomato plants. However, the studies have also shown that a virus is needed in order to confer the heat tolerance. So all three symbionts are required. And they know this because when they cured the fungus of the virus, both the fungus and the plant did not survive these high temperatures. So the objective of our study is to look at this three-way interaction involving the plant, the fungus, and the virus. So our hypothesis is the symbiotic interaction between the tomato plants, the selected virus, and the associated fungus will result in increasing plant tolerance to drought and high temperature. So we did 2 experiments, one dealing with drought conditions and one under heat stress conditions. In both, we used 3 treatments. The first is the non-symbiotic treatment, which would just be the tomato plants. The second would be virus-free, and they are the plants inoculated with the fungus, but not the virus. And the third would be AN or anastomosis, meaning that the plants are inoculated with both the fungus and the virus. Okay, so this picture shows the seedlings were germinated from seeds, and then in these beakers there are the spores of the fungus and the virus. And after they've grown a bit, we plant them in these plastic trays. And they were allowed to grow in the trays for 2 weeks before being transferred to the sterilized pots. Then we divided the plants in 2 groups, 2 stages, pre-anthesis and anthesis. Pre-anthesis means prior to flowering, and anthesis means during flowering. So the first table you see in my right-hand picture is going to be our pre-anthesis stage plants. And then the second table will be our anthesis. So we wait until we get to the pre-anthesis stage, then we take our physiological measurements, and then we do the same with the other group once they reach their stage of development. So the plants were allowed to grow under drought conditions, meaning we didn't water them, for 3 weeks, and then we took our physiological measurements, such as photosynthetic rate, stomatic conductance, water potential, and the photosynthetic pigments, as well as soluble sugar content. And this is just a picture of me taking the photosynthetic rate with the Lycor machine and working on the photosynthetic pigments on the lower left. Okay, moving on to the results. We also measured shoot and root growth. Between the stages, we found that there was no difference in the shoot and root growth between the treatments. The second result we found was with the photosynthetic rate, and we also did not see a significant difference, so the plants were not changing their photosynthetic rate under stress, or they were doing it equally. Okay, and the photosynthetic pigments, we measured were chlorophyll A, B, and carotenoids. And these are important because these are pigments found in the chloroplast, and their function is to capture light energy. So we wanted to see if the levels of these were changing, because under stress, chlorophyll tends to be degraded and other pigments take over. But we did not see any difference between the pre-anthesis and anthesis stages, and we did not see a difference between the treatments. Okay, and the other thing we measured was leaf water potential, which is basically measuring the water status in the plant. But we did not see a difference with this as well, so we did not see any sign of any osmotic adjustment between the treatments. The last thing we measured was soluble sugar content. And like I said, under stress, the plants either make osmotic adjustments through sending compatible solutes to the vacuole, which brings more water, or it doesn't. And the 3 treatments showed the same amount of solutes, so there doesn't appear to be any sort of adjustment. In conclusion, for the drought, we reject our hypothesis because we did not see any significant difference between the treatments. Well, before I move on to the heat, I do want to make it clear that just because we didn't see any significant differences doesn't mean that the research isn't important, because all research contributes to our knowledge. And so now farmers and investors won't invest all their money into something where studies have shown that it works, but our study does not show that. Okay, so moving on to the heat, we use the same 3 treatments, non-symbiotic with just the plant, virus-free with the fungus and the plant, and the AN with all 3. Each of these treatments received 4 samples and were placed in soil simulators, as you can see right here. And the control box has wires that run into the soil simulators, which control the soil temperature. So that's how we monitored the high temperature soil. And we also used temperatures in the soil just to make sure everything was the same. We also made sure that the water was supplied continuously through wicking ropes in the bottom of the tins that run down into the pots with water in them. So water was continuously supplied. And this shows a picture of the wilting process and chlorosis at the end of the experiment. Again, we measured photosynthetic rate, and we found the stomatic conductance was not affected between the treatments. However, there was a significant difference in the photosynthetic rate on a statistical level between the control plants and the virus-free and three-way plants. We can draw a conclusion from this that it is the fungus that is most important in this interaction and not so much having all three of them, the virus, because previous studies have said that you need the virus as well. But we can see that the fungus, the virus-free, is operating at the same photosynthetic rate as the three-way. Again, we measured the photosynthetic pigments. We did not see any significant difference in chlorophyll A, but we did in chlorophyll B, although the ratios stayed the same. Okay, this shows clear visual evidence of the three-way interaction. As you can see, the plants on the left are the non-symbiotic, which is just the tomato plants. And you can see clear signs of wilting in those plants and as well in the virus-free plants, whereas the three-way symbiotic plants look a lot more healthy. So in conclusion, we accept our alternate hypothesis because we did see improved results in our symbiotic plants. And it appears, like I said, that the most important interaction is between the fungus and the plant rather than the virus, the fungus, and the plant. And I would like to thank Dr. Morsi and his grad students, Hayden and Caitlin from West Alabama, for helping us grow our plants and inoculating them, and Jacksonville State University for all their support. Thank you.